Hello, everybody. Welcome to this live webinar. Thank you for joining us today. I hope you can hear me loud and clear. I'm Michela Fassi, Communication Manager at Milestone, and I'll be your moderator for today's webinar. Today, we are here for our live webinar titled, Don't Trust Leaving Your Cassettes Alone, Track Them. And we will see together many issues and problems and, of course, solutions for traceability. Milestone is in the field since 30 years. Our mission is to reduce patient anxiety by providing pathologists with technologies for same-day diagnostic results. And we are proud to offer innovative solutions in different fields like tissue processing, sample handling, macro digital, tracking, frozen section, cytology, and reagents. Before we start, I'd like to focus your attention on this slide for a few seconds. The meeting controls hide when you not use them, and they reappear if you move your mouse. Your microphone will be muted for the entire webinar, but of course you can chat using the chat box to communicate anything with me. This webinar is intended to be interactive, so we encourage you to ask questions, and you can type your question anytime during the webinar in the Q&A box. There will be a live Q&A session at the end of this presentation. Now let's see together the speaker of today. The speaker today is Lisa Longaretti, application specialist at Milestone since more than 7 years. She has a degree in biomedical laboratory techniques, and she has great experience in histology research and biobanking. This webinar today will be recorded and will last about 40 minutes. Now I will stop my presentation and I will give the floor to Lisa in the laboratory. So Lisa, you have the floor.
Good morning and good afternoon to everybody, and welcome in the Milestone laboratory. Today we will talk about the traceability of samples. We will focus not only on the cassette used for sample processing, but on the entire process of the specimen. We will focus on the sample's traceability, following the specimen journey from the moment of its collection up to the final diagnosis. Traceability, you know, is a great value for any laboratory, with the precious goal of improving a patient safety. Given the frequency which some errors occur and their potential effect on patients, the rate of specimen identification errors may be an important measure of patient safety. As you well know, each sample is unique and unrepeatable. Therefore, a mistake can cause harm to the patient. Traceability can also enhance the laboratory efficiency and optimize the workflow. In the daily laboratory routine, operators have to deal with several tasks and they often have limited time. For example, manual scanning of the cassettes is a time-consuming step that occupies valuable operator's time. And an increase in time means an increase in cost. Moreover, tracking cassettes in the laboratory can provide real-time detection of workflow bottleneck. If not properly tracked, there is high risk of losing specimens or having mismatches between specimen and documents. So nowadays, traceability is a really key factor to patient safety for monitoring and tracking the specimen. The sample makes a long journey, starting from the operating room through various steps up to the diagnosis made in the pathology lab. Pathology lab usually process a large volume of specimen on a daily basis. This specimen needs to be properly identified to ensure that patients receive the proper diagnosis. However, manual procedure can be time-consuming, and of course, there could be errors leading to misdiagnosis. To help address this issue, labs have started to adopt the STS, the Specimen Tracking System, that automates specimen tracking, thereby reducing error and improve efficiency. Before going into the details on tracking system, I'd like to focus on the terminology. The terms tracking and tracing are often used interchangeably, but in reality, they identify two mirror processes. Tracking means to follow the path of the sample from the starting point to where the sample is in this moment. On the other hand, tracing is to look at the complete history of the sample that I need to recover during the diagnosis. So we know that it's important to ensure the traceability of sample from the moment of the collection and during the working cycle up to the archiving. It's also essential to avoid identification error or losses, to ensure proper storage and custody and archival material in pathology lab. I think you will agree with me that tracking has become an integral part of our life and cover almost every aspect of our private and working life. Nowadays, we are used to have everything under control and to track everything. So this is something we should do properly also in our lab. And Milestone has developed a line of devices that cover most of the steps of the specimen journey, and also a software that allows to track and trace the specimen's data along this way. Before showing you the Milestone solution for traceability, I'd like to focus on each step of the specimen journey and see together which are the common issue and error with the traceability. Let's start from the collection of the specimen in the operating room. This is the first step of the specimen journey. Some very common problems are represented by the communication error and mislabeling during this step. Any error in verbal communication and in the transcription during the hands-off may increase the risk of mislabeling the specimen and mismatches between the specimen and its requisition form. Specifically and unfortunately, there are frequent errors in regards of specimen not labeled or labels with the details of wrong laterality. And to confirm what I have just explained to you, here's a study that shows the identification errors frequency of the surgical specimen. You can see the list of the number of errors that have been identified in the study, and the error frequency that is more than 4 per 1,000. So we have already detected several issues of traceability in the first step of specimen journey. And now let's move on the second step. Let's now consider the transfer of the specimen from the operating room to the pathology lab. Here, the method of transport must be guaranteed the traceability of the sample, the transport time, and its adequate conservation. In this regard, the hospital department should develop and implement a written or digital procedure for the correct mode of transport intra and extra hospital of the biological material. Upon acceptance in the pathology lab, the sample and the request are checked and a new internal anatomy code is assigned. This point also is crucial, as relabeling errors may occur here. And after that, let's move on the grossing phase. Here, it's necessary to perform a manual check between the data reported on the request and the data reported on the container. Check the patient identification data, check the tissue contained in the container, check the number of the containers. If one of these data does not coincide, it means a waste of time in search and delay in the diagnosis. During grossing, error may also occur during the preparation of the cassettes, where the patient codes are written manually or printed, but a different code from the patient code. After grossing, the residue of the sample must be placed in the archive. Here, errors of incorrect positioning of the sample in the archive can occur. with the consequent loss of time in case of sample search for resampling. Let's move forward. After grossing, the samples are processed and then transferred to the embedding station. At this stage, one or more cassettes may accidentally get lost. They may fall to the ground or may remain into the processor. And during the embedding, I could miss one or more biopsy because I don't know how many samples were contained in the cassette before opening. It often happens that small biopsies remain attached to the lid of the cassettes, which throw away, or hidden by residue of solid wax. And at the end of the embedding, a count of the cassette is carried out, which must correspond to the cassette inserted in the processor. This means waste of time in case one or more cassettes are missing. After the embedding, the paraffin blocks are cut at microton. In this stage, the block code must coincide with the slide code. If the slide has an incorrect code, it may be stained with wrong program, an example, with serious causes made for the patient diagnosis. When the slide is given to the pathologist for the diagnosis, it is essential not to lose any slide or block, because the consequence is always incomplete diagnosis, waiting for the patient, and waste of time for the laboratory staff. When the case is closed, slides and block will have to be filled in a correct position in the archive. And here again, errors in archiving can occur. So we have seen that errors in traceability may occur in each step of specimen journey. Also, we need to remember that every procedure affects the following one, thus creating a domino effect. Any error made in the previous step will have an impact in a final diagnosis. And so, how to solve all the traceability issues? The ideal solution is all-in-one traceability that is done both by software, by the tools that are used, starting from the operating room to the end of the entire pathological anatomy chain. An ideal tracking system should be automated to save the time for the operator and allow the specimen tracking system on the device. Numerous papers report that the deployment of a tracking solution is becoming common practice in pathology lab and research lab. These informatics tools enable automation, improve efficiency, facilitate traceability, and standardize workflow, and especially enhance patient safety. As you can see in this graph, there is a dramatic reduction of errors when tracking is applied. To intercept and avoid all these errors and to improve specimen traceability, Milestone has developed various systems, hardware and software, that cover most of the steps of the sample journey, with the consequence of saving cost and time and enhancing patient safety. We have focused on traceability on the pre-analytical phases, and we have developed a software that interacts with our tool, tracing the specimen data. And now I'll show you how it's possible. Here we are in the operating room. The sample is put into the bucket, and the ultra-safe device fills the bucket with the formalin in a closed system. During this operation, all data are saved and shown in the MileWatch software. At the end of the run, a label with all information is attached on the bucket, on the container. And all of these data are saved and shown in the MileWatch software. After that, I can also activate the temperature data logger card to record the time and the temperature of the transport. Also, in this step, all data are saved and shown in the MileWatch software. when you receive the sample in the pathology lab, you have to check what was sent from the operating room and what was received in the pathology lab. In this way, all data are saved and therefore also the date of the acceptance of the sample. Moreover, the time and the temperature during the transportation are saved and a report is generated. Through the use of our grossing station on which the MacroPath digital system is installed, it's possible to record the audio and video during the grossing. Now, in this time, I open my case and I start with the grossing. And during the grossing, I can take a picture. All of these information are saved in the MileWatch software. And also, anytime you can check, you can ask to the MileWatch software, and each case ID can be called up at any time to check how the sample took place. The sample now is put in the cassette, and the cassette produced during the grossing need to be scanned and before processing. And how it's possible? We use our R-tracker device to scan all cassettes in automatic way. In this way, full cassette traceability is guaranteed before processing. And through the real-time monitoring function, the number of cassettes are ready to process. A short focus or an automatic scanning. Our R-tracker device is an automatic scanner that provides 3-in-1 operation: scan, check, and track. The scanning operation is fast, simple, and automatic. In this way, the operator can save a great amount of time and have accurate documentation of the process. Here again, all data are saved in the MileWatch software. After the scanning, the batch is ready for processing, as also shown in the monitor behind me. And through how it's possible to continue the traceability also during the processing step. Through our processor and the MileWatch software, it's possible to have complete traceability during the processing phase. In particular, it's possible to view the real-time processing and the real-time step. And you can see the processing parameters for each process. And you can see also the details of the reagent used. It's possible also to see the time and the temperature for each phase during the processing. And through our traceability system, it's also possible to search for a specific cassette by looking for its case number. You will find in which process the cassette has been inserted and which program is in running and what stage it's in processing. And at the end of the processing, with R-tracker again, you can make a comparison of the cassettes. So it's possible to perform a comparison between the cassette inserted in the processor and those removed from the processor at the end of the run. to check that no cassettes have been lost. After that, now the cassettes are ready for the embedding. And by scanning a cassette in our Histodream embedding workstation, the picture of the cassettes taken during the grossing will appear on the screen. In this way, I can control the number and the type of sample that I'm about to embed to check if the processed sample corresponds to the one that has been grossed. Also, during the cutting step, I can intercept a potential mismatch by using the same method. To conclude the specimen journey, after microtomy and staining, the slides are finally ready for diagnosis and then delivered to the pathologist. If during the microscopic evaluation, the pathologist wants to review or check the history of the case, he can carry out the tracing of the case through our MileWatch software. When you search for a specific case ID, all details are shown and you can trace the complete specimen journey. You can see how it was collected and transported. You have the grossing details and you see how and when it was processed. The entire history is here. Summarizing how it's possible to connect all these systems through MileWatch software. MileWatch is the software that connects all the steps that we have just described. Thanks to MileWatch, your LIS can be integrated with all information that MileWatch save from all the instruments. MileWatch is also 24 hours, 7 days live monitoring system. Our software can send instant notification via email, phone call, SMS, and WhatsApp, so that the interested people are informed in real time in case of alarm or unexpected events. Operator can monitor the unit status and performances or download the reports in real time. And this can be very useful to improve productivity of the lab. And finally, with the search function, it's very easy to retrieve all information related to a single cassette or case ID and have the complete history of the specimen journey. I hope that my presentation has been clear and useful for you, and now I'm ready to answer your question.